Welcome to Brainstorm, where we give you a glimpse into the world of science for this Monday, March 26th, 2012. We begin with a story from the field of genetics. Researchers at Georgetown University have been studying a particular gene and found it does, in fact, have a direct connection to obesity. Interestingly, this gene isn't involved with metabolism, but actually in the development of synapses. This all started with mice that had a mutation to the BDNF gene, resulting in more immature synapses, affecting learning and memory. The mechanism behind this is relatively simple. After eating, the hormones insulin and leptin are produced and meant to be transmitted through neurons to the brain. Once there, the chemical messages trigger the suppression of appetite. So, a mutation prevents this signal, resulting in no appetite suppression and therefore obesity. Next was looking at BDNF in humans. Sure enough, large-scale genome studies found a strong link between obesity and variation to the gene. So now the question is how to correct this mutation. One option being gene therapy, but that has certain complications. Most likely, research will begin on finding a drug that stimulates proper production of BDNF. And from the world of medicine, scientists from Stanford University working with other institutions have created a more effective form of a protein used to treat certain cancers. This protein acts as a master regulator for the immune system, and injecting it triggers the growth of certain cells, including T cells, which can often be very effective treatment, especially against certain late-stage cancers when most other treatments fail. Despite its effectiveness, use of IL-2 is extremely limited due to severe side effects it causes. Lung cells have different receptors for the protein, so the high concentration required for a therapeutic effect causes pulmonary edema. In order to make a better form of this protein, many mutated forms were created and tested on their ability to bind to the receptors on T cells, resulting in a far more potent protein based on cell culture tests against certain cancers. The reason Super2 works is because binding to T cells involves 3 receptor stages. Regular IL-2 must bind to the alpha receptor and become the correct shape for the beta receptor, but alpha receptors are often very scarce, meaning high concentrations of IL-2 are needed. Super2, unfortunately, isn't less effective at triggering the edema, but its different shape allows it to be effective in smaller concentrations. Right now, the group that originally tested IL-2 is working with Super2, and it'll hopefully find clinical applications soon. Our final story is a quick update from the world of material science. A group has developed a way to make silk have strong antibacterial effects. As we've mentioned on Brainstorm before, silk is a pretty awesome material, with desirable mechanical properties and biocompatibility. Now with a simple chemical treatment, it can kill most bacteria, including anthrax. Bacteria from the species Bacillus can become dormant spores, allowing them to survive extremely harsh conditions, such as antibiotics and radiation. So oxidizing agents like chlorine compounds are often used to kill them. Silk is simply soaked in a solution similar to normal bleach and then dried. Testing this killed almost all E. coli placed on it after 10 minutes and similarly affected spores of an anthrax relative. This material could have a number of applications, including protective covering during a bioweapons attack and even water filters. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.